Hello friends. Have you ever wondered how the virus gets inside our body? Today, we will study about an entrance of virus inside the human body. Viruses are microscopic parasites, generally much smaller than bacteria. They lack the capacity to thrive and reproduce outside of a host body. All viruses have a nucleocapsid, composed of nucleic acid, surrounded by a protein coat, which is called as capsid. A protein coat, which function as a shell to protect the viral genome and helps in attachment of virus on the host cell. Some viruses have a membranous envelope that lies outside the capsid. Viruses can be classified based on their genome, that is, DNA or RNA, or other structural components, such as the capsid, the envelope, and the viral receptor proteins. Those viruses having an envelope are called enveloped virus. In these enveloped viruses, the nucleocapsid is surrounded by a lipid bilayer and glycoprotein, which is mainly derived from host cell membrane. Whereas non-enveloped virus, lacking an envelope outside the capsid, also called naked virus. The enveloped virus are easy to sterilize than non-enveloped virus, because the lipid envelope of these viruses is relatively sensitive to desiccation, heat, and soap. Enveloped virus mainly involves coronavirus and retrovirus, while poliovirus is a non-enveloped virus. Okay, this was about the viruses. Now, let's see how viruses enter the human body. As we know that the human cell has a covering made of double layer of lipids and proteins. Virus must transfer the lipid layers without killing the cell. But it cannot pass through skin, because the outer layer of human skin made of keratinized cells, called the stratum corneum. Virus cannot transport it across dead and keratinized cells. Due to these keratinized cells, on outer surface of the body, the virus must enter into the body through various means, as droplets containing viruses in the air, spread by coughing or sneezing, and enters the body through the mouth, eyes, nose, ears, or enters through any wound on the skin. It can remain viable on surfaces for hours to days, and may be able to enter the lungs directly when inhaled. After entering the body, the virus spreads to the back of the nasal passage and to mucous membranes in the throat, attaching to the body's cell receptors. As epithelial cells made up the linings of cavities in the body, such as the lungs, small intestine, and stomach. The modified epithelial cells, known as goblet cells, secretes mucus on the surface of the membrane and traps harmful substances such as viruses and bacteria on the surface cell. Depends upon the type of virus. There are basically 3 different ways of their entry into the host body, and each mode comprises a series of steps, which must be successfully complete to establish a productive infection. One of the 3 different modes of entry employed by most viruses is by the direct penetration of viral genetic material into host cell, which is basically followed by non-enveloped viruses or naked viruses. In this case, the virus come in contact with the host cell with the help of capsid protein outside the virus. After making contact with the host cell, the rearrangement in capsid protein allowing the viral genome to pass through the membrane of the host cell, and then it allows the genome to enter into the cytoplasm of cells. As poliovirus, which is a non-enveloped virus, binds to a receptor on the host cell surface, and then form a pore in the host cell membrane to inject genome inside the cell. After entering into the host cell, the virus hijacks the cell's protein synthesis machinery, causing inhibition of cellular, and initiates the synthesis of viral proteins, that is, multiple copies of viral genome. From these viral genome, number of viruses produced by each infected cell. After the formation of multiple copies of virus inside the host cell, the new virions produced, which are able to infect adjacent cells, and repeat the replication cycle. Some viruses are released when the host cell dies. However, other viruses can leave infected cells by budding through the membrane without directly killing the cell. Another mode of entry, employed by both enveloped and non-enveloped viruses, is receptor-mediated endocytosis. Receptor-mediated endocytosis, a process by which cells absorb metabolites, hormones, proteins, and in some cases, virus by the inward folding of the plasma membrane, which is also known as invagination. In this method, virus recognize and binds to the receptor present on the host cell membrane. After binding to the receptor, cell membrane starts folding inside, along with receptor, virus complex. On the completion of invagination, vesicles containing virus, now, detach from the cell membrane. Let's discuss, with an example of influenza virus. Influenza virus is an enveloped virus, first binds to the cell surface receptors, and induce receptor-mediated endocytosis. After detach from the cell membrane, the pH inside the vesicle starts decreasing, and this acidic environment inside the vesicle leads to the detachment of virus from the receptor. After taking off from the receptor, virus envelope fuses with the inner membrane of vesicle and start releasing nucleocapsid into the cytoplasm of host cell. However, in case of non-enveloped virus such as adenovirus, induces receptor-mediated endocytosis and then disrupts the vesicle membrane, releasing capsid into the cytoplasm. A number of viruses produces by each infected cell. After the formation of multiple copies of virus inside the host cell, the new virions are able to infect adjacent cells and repeat the replication cycle, and finally spread around by coughing, sneezing, and enters the body through the mouth, eyes, nose, ears, or enters through any wound on the skin. Now, let's see the third mode, that is, direct fusion with plasma membrane by which HIV enters in our body. This method mainly employed by HIV, a non-enveloped virus. In this method, virus makes a direct contact with the host cell membrane, with the help of their envelope. After making a contact, virus releases its genome inside the cytoplasm of host cell, along with capsid. After releasing from the capsid, viral genome replicates and form a multiple copies of virus inside the host cell, and the new virions are able to infect adjacent cells and repeat the replication cycle, and leads to various symptoms.